Hello viewers, I am Dr. Robiul. I work as a lecturer in pathology in a medical college hospital, and I am making this video for my students and also for you. Hope someone finds this helpful. Today we will talk about a very interesting topic, and that is why do we age and what are the mechanisms of aging. This video will contain the definition of cellular aging and the mechanisms of cellular aging, that will include the roles of DNA damage, cellular senescence, that will include the role of telomere attrition, and also the role of activating tumor suppressor genes. We will also discuss about the role of defective protein homeostasis and nutrient sensing in aging. Okay, so a lot of topics, so let's begin. First question, what do we mean by aging? Now always remember, old age is a concept of longevity in human beings. The consequences of aging usually appear after the reproductive age. Now, since most of my students in my medical college are very young, they are young medical students, they don't like to study unless I give them pressure, and they are very fond of movies. So I usually begin my discussion of cellular aging when I take their classes with the movie Pirates of Caribbean on Stranger Tides. Have you seen the movie? Yes, on that movie there was a fountain called the Fountain of Youth. And if you drink water from that fountain in a certain time, you will have eternal life and you will be youthful forever. Unfortunately, in reality, that type of fountain is nowhere to be found. So why do we age? Human beings age because the cells of our body age. And although whenever we talk about aging, we mainly focus on the cosmetic manifestations of aging, but always remember, aging has even more important health effects. For example, many of the chronic diseases, they occur in old age. So aging is one of the strongest independent risk factor of some chronic diseases like ischemic heart disease, Alzheimer's disease, cancer, etc. So how can we define cellular aging? Cellular aging can be defined as the result of progressive decline in cellular function and viability caused by genetic abnormalities and accumulation of cellular and molecular damage due to effects of exposure to exogenous influences. Okay, I hope you're still with me. You didn't run away just like my students do when I try to teach them definitions of pathology. I even have to show them teddy bears to keep them calm. So look, I'm also showing you teddy bear. So look at the teddy, don't run away, because I will explain this definition line by line now. So what did we see in the first line of the definition? Cellular aging is happening due to progressive decline or gradual decrease in the function and viability of the cell. So cellular aging is the result of progressive decline in the cellular function and viability. Now, why is this decline happening? Why are we having this progressive decline in the function and viability of our cells? It is caused by genetic abnormalities. And why will we have genetic abnormalities? It can be due to various reasons. It can be due to accumulation of cellular damage, molecular damage, and also due to exposure to exogenous influences. Okay, so now that we have defined cellular aging, now we will move on to the next topic and discuss the common mechanisms of cellular aging. So there are a lot of mechanisms that can result in cellular aging. They include DNA damage, cellular senescence, defective protein homeostasis, deregulated nutrient sensing, etc. So let's talk about these mechanisms one by one. So the first mechanism was by DNA damage. Now we all know all the cells of our body, most of them contain nucleus, and inside the nucleus we have DNA. Can you tell me one cell that doesn't have nucleus? Very good, yes, red blood cell. So that is a cell that doesn't have nucleus. So coming back to today's discussion, so in the nucleus we have DNA, and that DNA can get damaged by both endogenous agents and exogenous agents. Among the endogenous agents we have reactive oxygen species that can damage not only the nuclear DNA but also the DNAs that are present inside the mitochondria of the cell. Similarly, exogenous agents, either physical, chemical, or certain biologic agents, all these things can also damage the DNA. Now, most of the damaged DNA do get repaired. However, some damage persists, and as the cell ages, those damaged DNA begins to accumulate, and that results in the progressive decline in the function and viability of those cells. Now, in your textbook, you will see some examples of diseases that happen due to DNA damage and results in premature aging. So there is a disease called Werner syndrome, that is a disease where we will see premature aging, and the problem here in Werner syndrome is there is DNA damage, and that is resulting in defective production of helicase. Now, helicase was an enzyme that helped in DNA replication, repair, and also that thing was required for unwinding of the DNA. So all these things will now become hampered, and as the damaged enzyme cannot help in DNA replication, repair, and DNA unwinding, so more and more chromosomal damage will accumulate, and that will result in premature aging of the cells, and that will result in premature aging of the individual as well. The next mechanism of cellular aging is by cellular senescence. Now, what do we mean by cellular senescence? Well, always remember, all normal cells have a limited capacity to divide or a limited capacity for replication. That means after a fixed number of cell division or after a fixed number of replication, the cells will no longer continue to divide. They will become arrested in a non-dividing terminal state, and this is known as cellular senescence, or also sometimes called replicative senescence. So why will this thing occur? And in your textbook you will see that there are two mechanisms that can cause cellular senescence. One of them is telomere attrition, or shortening of the telomere, and the second mechanism of cellular senescence is by activating certain tumor suppressor genes. So we will now talk about these two mechanisms briefly. So first we have to know about telomere attrition, and in order to understand telomere attrition, first we have to know what is a telomere. Now always remember, telomeres are short repeated sequence of DNA. They are found at the ends of linear chromosome. So here I am showing you an image of a chromosome. You can see on the both ends of these linear chromosome we can see telomere. What is the function of telomere? The role of telomere is it helps in ensuring complete replication of the chromosome ends, and also telomere protects the chromosome ends from degradation and fusion. So telomere is very important for cell division and replication. But the problem is, every time a somatic cell divides, small segment of the telomere is not duplicated. Therefore, with every cell division, the telomere is becoming gradually shortened. So there will be one time when the telomere will be lost, and what will happen then? The ends of the chromosome will no longer have the protection that was provided by the telomere. So there will be problem. The chromosomes cannot be protected and there will be formation of broken DNA. And whenever these type of broken DNAs are produced, that will signal the cell cycle to stop or the cell cycle to arrest. Now the next thing that I will talk about is regarding an interesting enzyme called telomerase, and this is very important to understand telomere attrition. Now a common mistake my students do is whenever they hear the term telomerase, they think that it is an enzyme that's function is to break down telomere. But no, don't fall for that trap. As a matter of fact, the function of telomerase is to maintain the length of the telomere. And how does telomerase enzyme do this function? By addition of nucleotide at the ends of the chromosome. So always remember, telomerase is a RNA protein complex and it uses its own RNA as a template for adding nucleotides to the ends of the chromosome. Telomerase activity is seen in the germ cells. It is present at low level in stem cells, but unfortunately telomerase activity is absent in most of the somatic cells, and that's the reason why most of the somatic cells will undergo cellular senescence, and that will result in cellular aging and aging. Now you may be asking, Dr. Robiul, can't we reactivate telomerase? Well, this same thing happens in certain cells. Can you tell me in which cells telomerase activity is reactivated? In cancer cells. Now always remember, cancer cells are immortal. They have unlimited replicative capability. They can divide for unlimited number of time. And how do they do that? They achieve that by reactivating the enzyme telomerase, and by reactivation of the enzyme telomerase they can maintain the length of the telomere in those cells, and that can allow those cells to replicate or proliferate indefinitely. The next mechanism of cellular senescence is by activating certain tumor suppressor genes. Now one important locus that you have to remember is the CDKN2A locus, because this locus is responsible for encoding two important tumor suppressor genes, and one of them is known as P16 or INK4A. And the function of P16 is to control the progression of cell cycle. And if I want to be more specific, P16 controls progression of cell cycle from G1 phase to S phase. Now, what is the role of P16 here? Whenever there is risk of uncontrolled proliferation, P16 will protect the cell by pushing the cell away from the cell cycle towards the senescence pathway. So that is the role of tumor suppressor gene in cellular aging. Whenever there is risk of uncontrolled proliferation or uncontrolled mitogenic signal, the cells are pushed out of the cell cycle and towards the senescence pathway. So now that we have talked about the first two mechanisms of cellular aging, which were DNA damage and cellular senescence, now we will move on and talk about the third mechanism, and that was by defective protein homeostasis. Now always remember, protein homeostasis can be achieved by two mechanisms. The first mechanism tries to maintain the correctly folded conformations of the proteins, and this is mediated by chaperones. And the second mechanism of protein homeostasis is via degradation of the misfolded proteins. And the misfolded proteins can be degraded by two systems. One is called the autophagy lysosome system, where first there is formation of autophagosome, and the misfolded proteins are taken inside that autophagosome, and then lysosome comes and fuses with the autophagosome and forms the autophagolysosome, and then the enzymes are released from the lysosome and they degrade the misfolded proteins. So that is the first system to degrade misfolded proteins, and that was called the autophagy lysosome system. The second mechanism to degrade misfolded proteins is via the ubiquitin proteasome system. Well, what happens here? First, ubiquitin binds with the misfolded proteins and makes them the target for the proteasome system. Then the proteasome system degrades those ubiquitin-coated proteins. So whenever there are some abnormality in the protein homeostasis, that will hamper the function, the viability and replication of the cell, and that may lead to rapid cellular aging as well. And sometimes accumulation of misfolded proteins can also trigger the pathway of apoptosis. Now, I have a separate video entirely on apoptosis, and you can also look into that video after watching this for more information. The last mechanism that we will talk about regarding cellular aging is by deregulated nutrient sensing. Now, the concept here is eating less actually increases longevity, and caloric restriction increases lifespan. And now we will talk about the mechanism that are behind these two interesting findings. So first we have to talk about the insulin and insulin-like growth factor signaling pathway, and then we will talk about sirtuins. That is one type of protein, because these two things play a role in nutrient sensing, and then nutrient sensing also plays a role in cellular aging whenever that is not regulated properly. So insulin and insulin-like growth factor 1, these are produced in response to growth hormone secretion from the pituitary in various cell types. So first, the pituitary gland will secrete growth hormone. Those hormone via blood will go to distant site. It will go to different cells, and there the growth hormone will stimulate production of insulin-like growth factor 1. And as the name implies, insulin-like growth factor 1 mimics intracellular signaling mechanism of the insulin. And the main function of insulin-like growth factor is to inform the cell about the availability of glucose. And whenever it informs about the availability of glucose, that will promote the anabolic functions of the cell, and that will also promote growth and replication. And usually the insulin-like growth factor 1 signaling pathway has very important downstream targets. They include two important kinases. They are called AKT and mTOR, or mammalian target of rapamycin. The next thing that we need to know regarding nutrient sensing is about sirtuins. Now, sirtuins are NAD-dependent protein deacetylases. So these are one type of enzymes. And their function is to promote expression of certain genes and gene products of those specific genes will increase longevity of the cells. Now, how do they do that? They inhibit metabolic activity, they reduce apoptosis, they inhibit harmful effects of reactive oxygen species, and also they stimulate proper protein folding. And all these things result in increasing the longevity of the cell. So now you may be asking, Dr. Robil, why are you telling us about all these things? Why are you telling us about sirtuins and also about insulin-like growth factor 1? What are their roles in cellular aging? And the idea is, whenever there is caloric restriction, that increases longevity by two mechanisms, and they involve these two things. So one mechanism is by reducing the signaling intensity of insulin-like growth factor 1 pathway, and the second mechanism is by increasing sirtuins. Whenever there is reduced signaling intensity of the insulin-like growth factor 1 pathway, that will reduce the rate of cell growth and also the rate of metabolism. And all these things will also reduce cellular damage. At the same time, whenever we increase sirtuins, that will have two effects. Increased sirtuins will increase the DNA repair enzyme activity by deacylation, so that will reduce DNA damage and help in longevity of the cell. At the same time, the second way by which sirtuins will help is it will adapt the cell to the calorie restriction. So here, to summarize everything, I am showing you a flowchart where we can see all the causes of cellular aging and all the mechanisms that can counteract aging. So here we can see on the left side there is DNA damage causing mutation. Then after DNA damage we can see cellular senescence, decreasing cellular replication, and there is cell loss and also cellular aging. And then we can also see defective protein homeostasis, that is resulting in decreased cell function and aging. And also in the far right we can see some anti-aging mechanism, nutrient sensing, that can result in altered transcription that will increase DNA repair and also increase protein homeostasis and counteract aging. So this concludes today's video on aging and cellular aging. I hope this video was helpful. If you like my videos, do subscribe, comment, share, and let me know. And for my students, I will also like to recommend you to go through your textbooks to know more information. Okay, so that's all for today. Until next time, take care and stay blessed. Thank you.